So cancer is one of the negative effects that can lead from mutations. Cancer is something that is very difficult to define clinically, and that is because there's a large number of complex diseases that behave differently, and it depends on the cell types from which they originate. So somebody who has, say, lung cancer might have a very different phenotype than somebody who has breast cancer or uterine cancer from somebody who has maybe brain cancer. These cancers can vary in onset, age of onset, growth rates, invasiveness, prognosis, etc. However, all cancer cells are going to share two fundamental properties. The first is that they're going to have abnormal cell growth and division, or proliferation. Secondly, they will have defects in the normal restraints that will keep cells from spreading and colonizing other parts of the body, or metastasis. And you can have the first without the second. In the first, you can have abnormal cell growth and division, proliferation, and this would be what you would have if you had a benign tumor. However, if it has the ability to spread to other areas of your body, it can then metastasize. Cancer mostly is going to affect somatic cells, so only about 1% of cancer will affect germline, or due to germline mutations. And so most of these cancers are going to be newly arisen and not necessarily be something that is inherited. Cancers very rarely will arise from just single gene mutations, but instead it happens that you have mutations accumulated in many different genes, maybe 6 to 12 genes. Typically, these mutations are going to affect many different important cellular functions, things like DNA repair. Maybe if this is messed up, then the cell is going to start to accumulate more and more mutations. Problems with cell division. Maybe the cells are replicating abnormally and growing too much. Apoptosis, we'll talk about that in just a second, but maybe the cells are destined to commit cell suicide and they don't, but they stay alive instead. Cellular differentiation, maybe these cells act a little bit more like stem cells and they are able to continue to grow. They can exhibit migratory behavior instead of staying put. And loss of cell-cell contact inhibition is another characteristic of many of these cancer cells. So you can think of cancer as a multi-step process. Typically a single mutation not going to be sufficient to transform that normal cell into these tumor-forming or malignant cells. The incidence of cancer, and we see evidence for this because the incidence of cancer rises exponentially with age. And so it suggests that as time goes on, many independent mutations are required. If a single mutation was sufficient, you would expect the incidence of cancer to be independent with age. Second evidence is that there is a delay between exposure to carcinogens and the appearance of cancer. So again, it may cause some initial mutations, but you may need the accumulation of additional secondary mutations before it actually becomes a cancerous cell. Cancers themselves often will develop in progressive steps. You may start off with just some mildly aberrant cells, and it may progress and progress till the cells become increasingly tumorigenic and malignant. And ideally, we can start to identify tumors earlier. The person who has it identified much earlier is going to have a much better prognosis on average than somebody who doesn't have it diagnosed until it's much, much later. So cancer cells are going to contain genetic defects typically, things that are going to affect genomic stability, DNA repair, chromatin modifications. So we talked in the last lecture about xeroderma pigmentosum patients, and again, they're going to be defective in the nucleotide excision repair. So if they start to accumulate mutations, their body is not able to use this method to correct those problems. And so they're going to start to accumulate more and more mutations than somebody without this disorder. Another example in terms of chromatin modifications is epigenetics. In epigenetics, we look at modifications made to DNA, things like DNA methylation, and the patterns for the cancer cells are going to typically be altered compared to normal cells. And in fact, they are starting to now use these methylation profiles to diagnose tumors and help predict how severe they're going to be and how well they might react to certain different types of treatments. Overall, these cancer cells are going to have much less DNA methylation. However, promoters of some genes might be hypermethylated in these cancer cells. And so these cancer cells can't respond in a normal manner.